Hey guys, welcome once again. Um, so in the previous video, we studied how to call semantic variants using mutex tool, creating panel of normals. First, creating panel of normals and using the mutex tool, applying all the filters and all the QCs to be able to call semantic variants, right? Indels and SNVs, basically. Now the next thing we want to do is to do Germline short variants discovery, and I'm still at the GATK best practices website. Um, so what, how this works is that it takes a BAM file, and then you put this BAM file into this workflow, um, which basically calls uses a tool called haplotype caller to basically call the variants per sample, and then combines all the files together. It produces a GVCF when you insert it into haplotype column, and then you get a, GV, a GVCF, and then you use um, the process called joint calling to be able to kind of like call Germline variants for all the cohorts basically. Um, so in this case, which is good because it increases your power to be able to discover variants, and it helps to also remove false positive as well. Um, yeah, and then finally you get to VCF. You go through the process of filtering, and you evaluate your call sets, and you publish. You get some lucky genes. All right. So from here we could go. We could choose any of the workflows or the workspaces. Sorry, workspaces. It's actually workspaces. I mean, I think I like this. Like this. Yeah. Let's use HG 38. Or maybe for simplicity, let's use HG 19 or B37. This on Terra. You click, and then it will take you right. I'm going to use in Chrome. I'm going to use Safari. Alrighty. Just know what I was looking for. This is session before. Right, so we're going to click. Let's see what I opens. Okay, great. So I think this should work. Um, okay, all right. So this is the Germline XNP's workspace. You have some data already, comes with some data, one sample in the tables, and then there's a dashboard. So what we have to do first is to clone, right? And sorry, someone has to pay. Thanks, my supervisor, for support. So we are cloning, right? We are cloning to be able to. Yeah, so we've cloned the Gemline variants discovery workspace. We could edit. I think we won't edit at this time. And then we have the data associated with it. Let's see whether we have some notebooks. No. Then we have the first workflow. So I think it's numbered. So what you do first from also here, right, is to process for variant discovery, which is this one. Then the second one is haplotype color, which is this. This is joint scholar type. Joint discovery is this part, right? So since we've already You know, if you remember, we did something similar, sequence, conversion. So this should help us to get analysis ready, bomb files, analysis ready, reads, bomb files. This should help us get that. So there's no need of doing the first one. So you just zoom into the haplotype column. Okay. Oops. Okay. So this has to be removed. Um, which is interesting. So, wait, all right. So let's choose the HTTP. Let's see why it work. Sign in and sign in. Let's sign in again. So let's clone this. Use the HTTP workspace. Clone, right? Let's see whether the workflows work this time. Oops. Right. I mean, that's fine. So what we could do is to, okay. So basically what we could do, I mean, what we had to do is to send the inputs. All right, so we can edit. So, I mean, what we could do actually is to find a workflow, right? We could go to the doc store. Oh, sorry. We could go to the broad method record. Yeah, I mean, I have the type broad. So that's what. Okay. So now we have to cancel it. I mean, everything is already set, right? So if you clone the workspace, it comes with the reference data. Select your data sets, and then you just run the analysis. So make sure you check whether there are attributes here. Otherwise, it just writes into the files directory. It wouldn't be appended to your tables. So make sure the space is filled. Then you run, launch. Yeah. So that's what we did. So the third part is joint discovery. Let's see whether we have access to it. Yeah, I think for this we have access. It's already filled. If you want to change some of the variables, tune some of the parameters, I mean, that's fine depending on the type of work you're doing. and select your sample set. We want to delete our intermediary files and then run, right? Run. Okay, so launch it. Yeah, so that's it. I mean, after running, the results will be appended. You get a GVCF for each sample and the index. Um, yeah, and sample set you get after running the joins column, you get the VCF, which is the joint called samples and the index. That's cool. I mean, it's very cool once you know how to navigate the process. Yeah, thanks, and see you in my next video.